Hey, my name is Trang Nguyen.
And my name is Alison Lopez Orozco, and this is the infant antibiotic resistance to the gut microbiome.
So for the problem, microbiome coats every aspect of a newborn after the infants leave the mother's womb. The gut is known to house plenty of different microbes that can either be beneficial microorganism or pathogenic. Due to this, the gut microbes has been the center of research that the gut is linked to many diseases and disorder. Along with this, antibiotics have shown to cause changes in the human gut system, especially with infants. Right when the baby leaves the mother's womb, the infant is at a higher risk of contracting an infection, so doctors would prescribe multiple different antibiotics to lower the risk of infections. However, infecting multiple antibiotics will later lead to a bigger threat, a superbug, an antibiotic resistance bacterium. Next. A study done in 2017 focused on how administering different levels of antibiotics during the first 3 years of a children's life can affect adult life. The hypothesis centers around if the children were treated with antibiotics for the first 3 years of their life, then there would be an increase in antibiotics resistance compared to the children that did not administer any antibiotics at all. 39 children ranging from ages 2 months to 36 months were divided into groups of no antibiotics, 19 children, and a group with antibiotics, 20 children. 28 stool samples from each child was taken over a course of 3 years and placed through two types of sequencing. Whole genomes, shotguns, and 16S ribosomes RNA genes were used. This allows scientists to measure the antibiotics resistance stain located in each child. From this experiment, the gut biomes on non-antibiotic children and antibiotic children vary greatly. Strands of B. fragilis and E. rectal were barely found in antibiotic positive children, but antibiotic negative children had them. Also, the stability of gut microbiomes is influenced by antibiotics. Children have received dosage of antibiotics have decreased in gut stability, while children that did not receive dosage of antibiotics had a stable gut community. Not only that, but antibiotics resistant genes were sharply increased when antibiotic treatments were started, but when treatment stopped, there was not a sharp decline. Other studies have shown that early exposure to antibiotics can lead to wheezing and asthma in children. They suggest that decrease in bacterial diversity and microbiome can lead to an antibiotic asthma production impact. Next.
Okay, so the new research showed that 88% of people agree that antibiotic resistance happens as the human's body becomes immune to antibiotics. This is not entirely accurate. Resistance will happen within our body as the host environment for the bacteria. However, the essential difference is that the body's immune response does not alter. It is the bacteria in our bodies that alter. Approximately 99% of babies born with low birth weight obtain antibiotics during the first 2 days of their existence, which leads to antibiotic tolerance and the degradation of beneficial bacteria in the intestinal microbiota. During childbirth, antibiotics are given to the infants directly, altering the infant's intestinal microbiota. This causes a substantial increase in the antimicrobial resistant genes in infants. A report from the Science Translational Medicine explained that infants treated with early life antibiotics have lower microbial diversity and harbor antibiotic resistant genes. Researchers are also starting to understand that antibiotic exposures in early life and before birth might cause additional long-term health problems, including increased risk of infection, asthma, and obesity. The specific makeup of a baby's gut microbiome microbes is significant because it has affected their likelihood for contracting many baby and adult diseases.
And for the compare and contrast for the research article dealt with the effect of administering antibiotics during the critical lifespan of the child. The research was looking for antibiotic resistant strand that appeared in children that received treatments and children that did not receive the antibiotic treatments, such as the strands were B. fragilis and E. rectal strand found in antibiotic positive children.
And the news article dealt with the antibiotics that was administering the baby at low weight in children that are exposed to antibiotics before birth. Some children younger than 2 months of age have antibiotic genes even if they were never exposed to antibiotics. So in the similarities of both articles and the research, whether the infant was exposed to antibiotics before or after birth, it leads up to the adult disease such as obesity and asthma.
Next. And these are our references. Okay, so let me pause this. How